So last time we discussed the Hardy-Weinberg equation, which is a mathematical way of understanding population genetics. So if you have a population of oak trees or earthworms or eagles or whatever, and if the allele frequencies, the P and Q values, are stable, if they are not changing from one generation to the next, we can use Hardy-Weinberg equation to calculate everything we need to know about that population. So if we are given the frequency of, say, the recessive allele, we can calculate everything else mathematically. We can figure out the frequency of the dominant allele. We can figure out the frequency of homozygous dominant individuals within that population. We can figure out the frequency of heterozygous individuals, and we can figure out the frequency of homozygous recessive individuals. And you learned to do that through a series of Hardy-Weinberg problems that we worked through last time. But Hardy and Weinberg also produced a principle, sometimes called the Hardy-Weinberg theorem, or Hardy-Weinberg principle. And it's a way of better understanding all of the factors that contribute to evolutionary change within populations. We know from the work of Charles Darwin that natural selection is the driving force behind evolutionary change. But we also know that it's a little more complicated than Darwin could have imagined. Natural selection is not the only factor that contributes to evolutionary change. It is probably the dominant, most important factor, but it's not the only one. And so understanding the Hardy-Weinberg principle gives us a more clear picture of what's happening when populations evolve. So what Hardy and Weinberg did is they came up with 5 conditions, and they said if these 5 conditions are true, if they are met within a population, could be, like I said, population of oak trees in the forest, it could be earthworms, it could be ladybugs, it could be humans, whatever. If you have a population of organisms all interbreeding with each other, within the same species, if these 5 conditions are all met, then evolution will not occur. Allele frequencies, P and Q, will not change. The values will stay the same from one generation to the next. But that's only if all 5 of these conditions are met. So the first one is the population must be extremely large. How large is extremely? That's a little loosely defined. But basically, the larger the population, the less likely it is for allele frequencies to change. For evolution to not occur, there must be no migration. So if you've got, a population of squirrels on opposite sides of a river, there must be no point where squirrels are able to cross over from one side of the river to another and flow, so the genes can't flow back and forth between the two populations. Because if migration occurs from one population to another, new genes could be introduced. Genes could be swapped back and forth between the populations. There must be no mutations, no genetic changes, no new genes being created through the process of mutation. The mating must be completely random. Now that assumes that this is a sexually reproducing species, but if mating is taking place, it must be completely random. Every single member of that population has an equal chance of mating, and there must be no natural selection. So nothing in the natural environment is favoring one allele over another. Well, try to imagine a population of oak trees, of earthworms, of ladybugs, of eagles, any population where all 5 of these conditions are met simultaneously. And you can't, right? Because that's never going to happen. there's probably not any population on this planet where all 5 of these conditions are always being met. And what Hardy and Weinberg are saying is that basically evolution has to occur. It's not a matter of is it going to occur, is it not? It has to occur. Because if any one of these 5 are not true, then evolution will occur. Allele frequencies will change. And so, you say, well, if this is just all completely hypothetical, if there's no such population where all 5 conditions are met, then why do we even talk about this? Why are we learning about this? Well, it's a way of better understanding the factors that affect evolutionary change. So if you have a population and it's evolving, like you can see that allele frequencies are changing, some genes are becoming more common and some are becoming less common over time, you can really dig deeper now and say, well, let's figure out which of these 5 things are not being upheld. And that'll help us understand what's really driving the evolutionary change within that population. So let's go through them one at a time. Well, actually, we'll do that in a minute. I forgot about this slide. Okay, so just to recap, Hardy and Weinberg are saying in their famous theorem, if any of these 5 conditions, if any one of the 5 is not met, if it is violated, then allele frequencies will change, and evolution, therefore, will occur. So a violation of any one of the 5 conditions causes Hardy-Weinberg equilibrium to be disrupted. Now let me explain. Hardy-Weinberg equilibrium is when P and Q values are locked in. They are not changing from one generation to the next. But if you violate any one of the 5 conditions, then that equilibrium will be disrupted. Allele frequencies will change. And by definition, evolution has occurred. We talked about this in the previous lesson, in lesson 3A, that evolution can be defined as any change in allele frequencies from one generation to the next. If P and Q values are changing, then evolution is occurring. Okay, so we're going to go back through these 5 conditions. We're going to go through them one at a time, and we're going to see how a violation of that one condition might contribute to evolutionary change. And we're going to start with number 1, which says that the population must be extremely large. Now, why does it matter, the size of the population? For this reason. If a population is small, like if we're talking about a population of 10 crickets in a grassy field, that would be small compared to, say, 5,000 crickets in a large field. If the population is small, allele frequencies are likely to fluctuate unpredictably and randomly from one generation to the next due to a phenomenon known as genetic drift. Genetic drift is defined as a process in which allele frequencies change unpredictably and randomly from one generation to the next within a small population. Let's look at an example of genetic drift. So here we have a very small population of wildflowers. You can see that most of them are red. There are some homozygous, well, there's 1/10 of the population is homozygous recessive and has white flowers. So in this small population of 10 plants, let's suppose that 5 of them, just by random chance, happen to be growing in a position where they're getting plenty of sunlight, plenty of nutrients from the soil, and the other half of the population maybe are growing in an area that's too shady, or maybe it's rocky soil, so they're not getting enough nutrients. So it's not about natural selection favoring a certain color, like the red surviving better than the white or vice versa. This is just chance. This is just random chance. Let's say that only 5 of the 10 plants are able to survive long enough to release their pollen and mate. So just by chance, even though the white flower is only 1/10 of the whole population, if that white plant just happens to be one of the lucky 5 that's growing out in the sunny spot where there's plenty of nutrients in the soil, and so therefore this is one of the lucky 5 that actually gets to reproduce, then the next generation is probably going to have a higher percentage of white flowers and certainly a higher percentage of recessive alleles. Notice the value of Q went from 0.3 up to 0.5. That's not because natural selection favored white flowers over red. Remember, it was just luck. It was just where the seed happened to fall and where the plant happened to sprout. These 5, the highlighted 5, just happened to be the ones that were growing in a place where they could survive. And so within this very small population, we could have a relatively fast shift in allele frequencies over just one generation, just by chance. That's what genetic drift is. Let's say it happens again, only this time only 2 of the plants happen to survive and reproduce. Again, just by chance. So now it turns out that the 2 that reproduce are both homozygous for the red allele. Not because natural selection is favoring red over white, but just by chance. And look at what happens. Now we've got big R mating with big R. None of the other plants are successfully breeding just by chance. And so what do you end up with? A population where the gene pool has shifted so that the allele frequency of the red allele is 1. 100% of the gene pool now is big R, and the frequency of the recessive allele has gone to 0. That is over just 3 generations, rapid change in the value of P and Q just by random chance. That is what genetic drift is. Now, that's not likely to happen if this original population was, say, 10,000 plants, right? These shifts in P and Q just due to random chance generally only happens in really small populations. Now, there's a couple of special circumstances where genetic drift occurs. One is called the bottleneck effect, where you have a really large population with a decent amount of genetic diversity, but then something happens to that population where it almost goes extinct, but not quite. Like the numbers die off drastically, but there's a few survivors, and in that surviving population, there's not going to be nearly as much genetic diversity as there was in the original large population. So let's take a closer look. Here we're using just a visual representation, a large bottle of hundreds and hundreds of marbles. Those represent different alleles in the gene pool. Notice we've got a lot of blue, a lot of white, and a few of these yellows. So that's different alleles, different genes or genotypes in the gene pool. But if this large population goes through a near extinction event, it would be like taking that bottle, turning it upside down, and shaking out just a few marbles. So instead of hundreds or thousands, now our population has died down to only, 6 survivors? In this small surviving population, are we going to have the same genetic diversity that we had in the original population? Almost certainly not. Notice none of the yellow marbles happened to make it through that little bottleneck. So what does that mean? It means when a population's numbers go from big to small, it almost always loses some of its genetic diversity just by chance. It wasn't that natural selection didn't favor the yellow allele. It's just by chance, those few survivors that didn't go extinct, just none of them happened to be the yellow ones, just by chance. So that's called a population bottleneck. The small population after the bottleneck has much less genetic diversity and very different allele frequencies than the original population. And so here's some examples of populations, real populations that we know have definitely lost a lot of their genetic diversity due to population bottlenecks that occurred in the past. African cheetahs, that population almost went completely extinct just a few thousand years ago and has sort of rebounded, although I think it's still kind of considered an endangered species, but very little genetic diversity. The Florida panther and the Illinois greater prairie chicken, all three of these are populations that have very little genetic diversity due to near extinction events. Now, another very special case where genetic drift plays a big role in allele frequency changes is something called the founder effect. And this is where a few individuals from a very large population, a few individuals splinter off and start a new population, perhaps in a remote location or a separate location from the main starting population. And when you pull out just a few individuals, it's kind of like if you have a shuffled deck of cards. 1/4 of the cards are diamonds, 1/4 are hearts, 1/4 spades, and 1/4 clubs. But if you shuffle that deck of cards and you only deal 4 cards to somebody, are they definitely going to get 1 diamond, 1 heart, 1 club, 1 spade? No. In fact, they probably won't, right? That little small population of 4 cards is not going to have the same level of genetic diversity that the original deck of cards had. And so when individuals come out of a large population, those few individuals don't have the genetic diversity. They don't have the same values of P and Q as the original population. So let me help you by giving you an example. This is a great human example, a disease called Ellis-Van Creveld syndrome, which is extremely rare. It causes, among other, I think it causes dwarfism for one, but it also causes, among other things, extra fingers and toes to form on the hands and feet. So it's a form of polydactyly, extra fingers and toes. And it's very rare. So if you were to look in the German population, like all of Germany, millions and millions of German people, the frequency of the recessive allele that causes Ellis-Van Creveld is 0.003. Like it's almost zero. It's so rare. Well, in 1744, a small group of German people left Germany, sailed across the Atlantic, and settled in America in what is now Pennsylvania, and founded what we know today as the Amish population. So I don't know how many Amish there are today, but it started out as 200 people from Germany, and we know what year this happened. It was 1744. Well, it just so happened that among those 200, out of millions of Germans, in that small group of 200 people, among them were a man named Samuel King and his wife. I'm not sure what her name was, but those two people both happened to be carriers, just by chance. They didn't know they were carriers, but they just happened to both be carriers of Ellis-Van Creveld syndrome. Some of their kids were therefore born with this syndrome, and several of their kids most likely were carriers. And so you have what started out as a very small population, and just by chance, the allele frequency for this disease was much higher amongst these 200 people than it is in the German population as a whole. So even today, if you go into the Pennsylvania Amish population and you calculate allele frequencies, you'll find that the current allele frequency for Ellis Van Creveld is still pretty rare. It's only 0.07. What is that, 7% of the gene pool? But that is 23 times higher than the German population. 0.003 compared to 0.07. It doesn't seem like it, but that 0.07 is 23 times higher than the German allele frequency. And here is an Amish mother and her child who has inherited Ellis Van Creveld. You can see 6 toes and 6 fingers. So a great example of the founder effect, where allele frequencies can be radically different just because the population size is small. The second Hardy-Weinberg condition was that there must be no gene flow. There must be no alleles moving into or out of the population due to migration, due to the movement of fertile individuals or their gametes. Like we could be talking about plants on opposite sides of a river, but the pollen could blow, across in the wind, and so that would be a gene flow. So if new individuals or their pollen are moving into or out of a population, the allele frequencies within that population will change. So how does this happen? Well, insects or wind might carry pollen from, say, some white-flowered plants, and that pollen might be carried over into a population of mostly red-flowered plants. So automatically, the frequency of the white allele would go up, right, if that happens. Songbirds from Europe are known to occasionally fly over to the Dutch island of, I'm not sure how to say that, Vlieland, and they mate with the local bird populations on the island. Well, the genes that the mainland birds are bringing in are genes that are not well suited for survival on the island. And so what's happening is this is reducing the survivability. It's changing the allele frequencies in the island bird population in ways that are not good. It's making the birds less well adapted for survival on that island. It's sort of putting that island population at risk. Obviously, allele frequencies P and Q are going to change if new alleles, different alleles, are being brought in by individuals immigrating from another population. So therefore, if evolution is not going to happen, if we're not going to change P and Q, there cannot be any gene flow. The population must be really large, and there must be no gene flow, and there must be, condition number 3, there must be no mutations. What is a mutation? You know this. It's a random change in the nucleotide sequence of an organism's DNA. Why do mutations matter? How do mutations contribute to evolutionary change? Because mutations automatically and instantly alter allele frequencies. Sometimes it might be a recessive allele mutating to become a dominant allele, right? So the Q is going to go down, the P is going to go up. Or it could be the other way around. Or sometimes mutations do something really radical. Sometimes a mutation creates a brand new allele that has never existed within that gene pool. And then natural selection can sort out, what happens to that new allele. Either it's going to be favored or it's going to be selected out. But mutations create alleles and automatically alter, at least in a minor way, they alter allele frequencies. So, for evolution to not occur, there must be no mutations. All right, Hardy-Weinberg condition number 4. for evolution to not occur. The mating must be completely random. If individuals tend to mate within a subset of the population, such as their nearest neighbors or their close relatives, then random mixing of gametes is not occurring. And that's going to cause allele frequencies to change. Now, just by definition, mating can never be truly random unless each and every member of a population has an equal chance of reproducing. Non-random mating increases the frequency of some alleles while decreasing the frequency of others. So if the mating is not truly random, P and Q values will change. Now, you think, well, why would it not be random? You know, what factors might prevent the mating from being truly random? And I can give you two really good examples. The first is called dominance hierarchies, which is kind of an odd term. So what does that mean? Well, think about a pride of lions. We're all familiar. You don't have to be an expert zoologist to know a little bit about how lions do things. We've all seen The Lion King, for crying out loud, and we've all seen nature programs on Discovery Channel or something like that. So when you see a pride of lions, what do you have? Well, you have usually several females, and they do most of the hunting for the group. You'll have some of their cubs. And you may have some juvenile males kind of lurking around the perimeter trying to get a little food from the group. But there's only one dominant male, and guess who gets to mate? That dominant male has mating privileges with all of the females in the pride. Any other males that are lurking around the perimeter of the pride, they don't get to mate, or they had better not try, or they will get their butts kicked, right? So that's not random, right? Not all of those lions have an equal chance of mating. Only the one most dominant, and he's not shy about showing that he's the dominant male, only he is getting to mate as far as the male portion of the population. So that's a dominance hierarchy. There's a clearly established sort of pecking order of who's in charge, who's getting mating privileges, and who is not. You see something very similar to that amongst African gorillas, where you have multiple females in what's called a harem, and there may be several males, but only the most dominant sort of senior male, which we often refer to as a silverback. Only that dominant silverback is actually mating with the females. So the mating's not random. You know, his genes are getting passed to the next generation, but the other males in the group are not getting any chance to pass their genes down. So therefore, whatever alleles he is carrying, and of course the females, those are the alleles that are going to get passed down. And the alleles that the other males may have, those are not getting passed to the next generation. Another factor that can block mating from being completely random is something called sexual selection. So sexual selection virtually ensures that mating is not random. Sexual selection is a process in which individuals with certain inherited characteristics are more likely than other individuals within that same population to mate. And it very often, at least sometimes, has to do with the preference of the female and who she chooses to mate with. So there's two ways sexual selection can occur. The first is called intra-sexual selection. This is where males compete against other males for the privilege of mating. So you see this with deer. The bucks use their big antlers to kind of lock with each other and wrestle kind of and sort of demonstrate who's the most dominant male, and the winner gets the privilege of mating, and the one that's less dominant either has to go off and find some other group of females to mate with, or he just doesn't get to mate, right? So that's intra-sexual selection. But the more interesting form that this can take on is called intersexual selection. This is where individuals of one sex, it's usually the females, are very choosy in selecting their mates from the other sex. So female songbirds, for example, will generally only mate and only show interest in mating with the brightest colored males. It's not just coincidence that male cardinals are bright, bright, bright red. It's because the ones that are not don't get to mate. The females are only interested in the ones that are very brightly colored. Male lions have this enormous, thick mane. Why do male lions have a thick mane? Because the males whose mane is thin and wispy don't get to mate. The females have no interest in them. And intersexual selection can create some really bizarre results, things that would never be favored by natural selection. And can also produce really striking differences between how the males and the females look within the same species. We call this phenomenon sexual dimorphism. It's the differences in how the males and the females look. So somewhere I've got some good examples of that. So the male peacock has this enormous tail plumage, which he loves to show off to the females. Females, of course, do not have this, the peahen does not have this same plumage. We talked about the brightly colored male cardinal, much more bright than the female. And again, this is sexual selection. This is because she is not going to be interested in mating with him unless he impresses her with his bright color. So any male cardinal who doesn't quite have that showy red color is never going to get to mate. If you think about tail plumage on a peacock, natural selection would never favor that. He has to drag this long tail around when he's looking for food, pecking the ground, walking around the vegetation looking for food. He's constantly dragging around this large tail that makes him very prone to being caught by predators. So natural selection would not favor that at all. So the fact that he has this is because of sexual selection. It's because without the fancy tail plumage, peacocks don't get to mate. And so only the genes for the biggest, most showy plumage are getting passed down to the next generation. Mating is not random if females are choosing very specific traits that affect whether they're willing to mate or not. If only certain males are getting to mate based on their characteristics, that by definition is not random mating. Lastly, Hardy and Weinberg said for evolution to not occur, there must be no natural selection. If natural selection does occur, some alleles, the ones that increase the probability of survival and reproductive success, those alleles will be passed down to the next generation in a much higher proportion than the alleles that don't increase survival and reproduction. So, we could spend all day talking about examples of where natural selection is clearly occurring, but here's just a simple one. So Drosophila melanogaster, which is the common fruit fly, the same species that T.H. Morgan used for his genetics research, samples of the Drosophila population were done in the early 1930s. And they found that there was an allele, a very, very rare allele, that made fruit flies, some fruit flies, resistant to a insecticide chemical called DDT. So it was a gene called DDTR, resistance to DDT, and any fruit fly that inherited this extremely rare allele would not be killed by this particular insecticide. Now, the frequency for that allele was almost zero in 1930. It was like 0.000001 or something like that. Well, from the 1940s, the 1950s, and the 1960s, so about 30 years, DDT was heavily sprayed all across America, not necessarily targeting fruit flies, but mainly to try to control the mosquito population. But because DDT was being sprayed so rampantly throughout the environment, lots of fruit flies died. The insecticide killed a lot of the fruit fly population. But of course, those very few who happened to have the DDT resistance gene, they didn't get killed, they survived. And they mated with other flies that also had the DDT resistance gene. And so you better believe what happened over the course of that 30 years, the frequency of the DDTR gene went way up from 0.0001 to 0.37. So that by 1961, 37% of the gene pool was DDT resistance. So, the frequency of this allele was dramatically increased by natural selection because the environment, in this case, a human activity, spraying this insecticide, we humans have a knack for changing the environmental conditions, don't we? That human behavior of spraying DDT created a situation where natural selection favored one allele over another. For many generations, those flies without the DDTR gene were dying and not reproducing. But during that 30-year period, the flies that they just happened to have the DDTR gene, they were surviving. And they were living longer lives, and they were reproducing more successfully and passing down that DDTR resistance gene. So again, Hardy and Weinberg said, if all 5 of the conditions are met, let's go back and look at those again. If all 5 of these conditions are met, then P and Q values are not going to change from generation to generation to generation. Therefore, if all 5 conditions are met, evolution is not occurring. That we know that's never going to happen. we know allele frequencies are going to change, and this just helps us figure out where to look to try to understand what is making those allele frequencies change. Is it just straight up natural selection, as in the case of the spraying DDT and favoring the DDT resistance gene? Or are mutations playing a role, creating new alleles? Maybe new genes are being brought in by animals migrating into a population. Maybe it's just that the population is really, really small, and so genetic drift is occurring. Random changes happening just because the population is small. This tells us where to look, because if a population is evolving, one of these 5, or maybe several of these 5 conditions must be being violated. And we can start to try to look and figure out what's going on. Before I leave you today, I want to throw out an extra credit opportunity. I'll put that up on the screen now. Here you have it. Take a look at the screen. The first 10 of you to email me with this information, all 3 of these bullet points, will receive some extra credit. Just put extra credit in the subject line and hit me up with this information. Guys, hope you have a great day. See you soon.